Hi, everyone. Thanks for participating. It's my pleasure to host this panel on heterogeneous treatment effects. We have a bunch of great panelists here, and to get ourselves started, I'm going to ask each of them to give a 2 to 3 minute introduction about themselves, what they work on, and their perspective of which they look at this problem. To start off, let's start with David Sontag.
Hi, everyone. Thanks for having me on this panel on this very important and exciting topic. So I work on machine learning in healthcare, broadly speaking. But when I started to get into the area about 10 years ago, turned out that most of the questions I wanted to answer were causal questions. I knew nothing about causality at the time, and I started learning here and there by talking to my friends in other areas, economics, political science, and so on. And I recognized that there was a really interesting opportunity to try to bring machine learning techniques into an area which has been traditionally studied in these other fields, and started working in the field over the last few years. I'm really interested in questions about how can we relax some of the assumptions you need in order to do causal inference from observational data, questions about overlap, hidden confounding. I'm also really interested in how one can reduce these sample efficient or improve the sample efficiency of learning. So what do you do when you have a very small amount of data and very high dimensional feature sets for causal inference? And then also I'm really interested in the question of how one can increase or build trust in models before you would deploy it. And most recently have been looking at that in the context of off-policy reinforcement learning.
Thank you, David. I guess we'll take David Kent now.
So my name is David Kent. I work at Tufts Medical Center, and really my primary professional identity is really as a simple country doctor who's interested in prediction. I still work as a hospitalist in the hospital, seeing patients on the general medical ward. I do that about 20% of my time, and then the rest of my time I split between kind of running a graduate program, mostly for physicians in clinical research, and I teach prediction in that graduate program, and running a small research center, which we call Predictive Analytics and Comparative Effectiveness, PACE for short. And I guess that center title gives away the interest. I've been interested in kind of personalizing treatment effects and predicting treatment effects for maybe around 20 years. And I think the interest really emerged from my experience just as a physician, seeing patients. I graduated med school in 1991. I think that was the year that Gordon Guyot first coined the term evidence-based medicine. And my interest in this area kind of emerged from a sense of dissatisfaction or frustration with the way we were taught in the 90s how to apply evidence from trials, which was basically, you know, if the patient in front of you meets the inclusion criteria of a trial or the entry criteria of a trial, then it's likely that that overall effect likely applies to that patient. But you know, of course, we've been arguing really that you know, estimating an average treatment effect from a large group of patients is just a very different thing than deciding what's best for an individual patient in front of you. And so over the last, I don't know, maybe 20 years, I've been kind of looking at pretty standard regression approaches to analyzing clinical trials. And I've stuck with RCTs alone because, you know, even just doing modeling on RCTs really causes a lot of credibility issues in terms of applying the work clinically. And so doing things in observational data, I think, is combining both heterogeneous treatment effects and deconfounding in observational data is you know, still more problematic, but that's an area definitely that I'm interested in now. I'll just say, by way of summing up, that over the last couple of decades, you know, I can't say I've made all that much progress, except I think I understand better and better all the serious barriers to really trying to estimate or predict, rather, individual treatment effects. And my sense is that there's no unique or perfect solution to all the different problems we have. And that, it's kind of like the conflict in the Middle East. If you think you have a solution, you probably don't fully understand the problem. So I'm not going to, easily be dislodged from that prior bias that really there are no unique solutions. There are a lot of trade-offs when one method is compared to another. And I guess I'll stop there.
Thanks. How about you, Isa?
Hi, I'm Isa. I'm based at Brown University. You could say I'm an epidemiologist, though my initial training was also as a physician, and I actually was a student of David Kent about 10 years ago. My research interests focus on drawing causal inferences from diverse sources of data. This sometimes takes the form of meta-analysis, sometimes takes the form of assessing heterogeneity in observational data, and sometimes takes the form of generalizability or transportability analysis, where you take predictive or causal models from one setting and you want to see them operate in a different one. Over the past 5 years, my main interest has been this generalizability analysis, in which heterogeneity of treatment effects appears mostly as a nuisance. So it's something we have to model in order to estimate some bigger population functional. But sometimes I'm directly interested in the heterogeneity of treatment effect measure as an object of primary interest. I'd like to maybe start with a claim that may be intriguing to the first David, and I'm guessing the other panelists. And the claim will be that the reason people think there is no right solution is because the problem is relatively ill-defined. And that if we are very careful and we drill down and we define the problems carefully, often a productive melding of statistics and machine learning and causal methods in some cases can lead to very serious, efficient methods.
And finally, Suchi.
Thanks for having me. Very excited to be here. So to summarize my introduction, I actually accidentally got into healthcare about 12 years ago. This is when we were first starting to pull data out of Stanford's electronic health record, not knowing how complicated pulling data out of an EHR is. Since then, have had a roller coaster whirlwind of working at the intersection of machine learning and healthcare. And at a very, very high level, both my work at Hopkins at the lab, a research institute I direct, and then also a company we've spun out of Hopkins, is all at we think of it as twofold. On the one hand, it's defining problem areas in medicine, where the fact that we have all this data available now, if we can integrate this data intelligently, how can we drive real-time tools to inform or transform care delivery to improve patient outcomes? And so that's sort of working backward from the problem to identify what are areas where now the fact that previously data used to not be available, but now that it is, how does that change? Like, what are novel application areas where this data can now change the way we practice medicine? So that's one half of my all of our efforts. And then the other half is all in the methodological front. And on the methodological front, very broadly at the intersection of machine learning, causal inference, and probabilistic reasoning. And the reason, and in particular, I think instead of kind of doing a laundry list, I'll sort of talk about a couple of areas that I think are particularly pertinent to the panel. So first is, in medicine, there's been a tremendous excitement for predictive models. But one of the areas in the last 4 or 5 years we've been working on is that turns out predictive models, the way we used to historically define it, the challenge with it is they're not robust as you take them from one setting to another. They're not transportable. So really the way people were defining predictive models all along, it's what Issa said, the question itself, the objective function itself is ill-defined or incorrect. And so we've been sort of working on this notion of what I call reliable decision support. And so reliable decision support are models that are transportable, and they're motivated by building into the objective function more, bringing in ideas from causal inference. The objective is defined with more grounded causal observations about what is or isn't true about your environment, what you expect to be same, what you expect to be different and changing, and then therefore you can give guarantees of robustness in your model. So that's one thread. The second thread that's relevant is around treatment effects, which is as soon as you start doing any kind of estimation from these kinds of large-scale observational data, and you start bringing in ideas of causal inference, it's really important to understand notions of like when you can estimate things and what kind of confounders exist that impact the quality of your estimation. And one that's very principally true when you're starting to work with large-scale health data or clinical data is that the data are very sparse and irregularly sampled, and there are many, many different missingness assumptions there. So missing not at random, missing at random, and in many times when we do modeling, it's very common practice to take this data and kind of bin the data into simple, in computer science, we have this notion of Markov decision processes. And as soon as we start doing discretization, we introduce all sorts of confounding because you're losing information. So we've done a number of works on trying to think about continuous time models or models where we take into account the natural sampling granularity at which the data is sampled, and build models that are more robust again, and principally that's partly done by bringing in ideas from causal inference and also avoiding binning, which is sort of a natural temptation to do. So I think that's mostly it, and happy to, and one actually last third thread about transportability on models. All of the above obviously are very crucial for sort of getting users excited. And so as we've built and deployed models and practice where physicians are using it, it's been an area sort of where I'm increasingly putting in more time is to understand kind of what fosters engagement, what fosters adoption, what fosters trustworthiness. But that's sort of a much younger, I think, emerging area where it's less obvious what's, what the problems are.
Great, thanks everyone. So to get ourselves started, I'm going to ask a couple of questions. If you all out there have questions, I'll ask them as they come in. But to start, to everybody, what's like the biggest roadblock to you for achieving like heterogeneous treatment effect estimation in medicine? We heard a little bit about it from everybody actually already, but if you want to just answer that more directly, I'll take it. And unmute yourself in any order.
So I'll hazard a first try. And, I'll start with what Sushi said, and that's, basically all the problems that we have with outcome prediction apply also to predicting treatment effects. So if we can't predict outcomes, there's no way we can predict treatment effect. But then treatment effect prediction has a lot of problems on top of predicting outcome. It's just much, much harder. And I would say there are kind of, I don't know how I enumerate them, but maybe 6 barriers that are more unique to effect prediction than outcome prediction. And the first one is one that all the panelists kind of hinted at, and it's just the fact that individual treatment effects are just unobservable. And so unlike outcomes, which you can observe in individual patients, you can't observe the treatment effects in individual patients. Second problem is the problem of scale. You need the absolute treatment effect in order to make decisions on patients, but it seems to me, at least, that there's some empirical evidence that relative treatment effects are just more stable and transportable. And so that creates a barrier. The second, at least in a regression framework, you have problems with much bigger problems with power than you do when you're predicting outcome. You need many, many more fold the patients to predict with similar precision. And you also have much less prior information than you do with predicting outcome. We understand risk factors in medicine pretty well. There's a lot of predictive models that have already been done. Thousands and thousands of predictive models have already been done. We understand very little about effect modifiers. And so if the data is sparse to begin with, and you don't have external knowledge to help you along, you're really in a troubling place, right? So you have noisy data plus weak theory, and that's just an engine for problems. And lastly, the issue with treatment effects is you need to predict something much more precisely than you need to predict outcome. So in general, treatment effects are just smaller than outcome probabilities, right? Because treatment effects, you know, maybe a typical treatment effect is a 20% relative risk reduction, so it's 1/5 of an outcome rate. And so if you have error in your prediction of the outcome in the treatment arm, error in your prediction of the outcome in the control arm, in some patients those errors compound, so you get error compounding, and then you get error magnification, because what you're interested in is something that's really precise. And so the imprecision is much more important and much more influential in terms of making decisions on individual patients. So those are kind of the half dozen things that I think make effect prediction more difficult than outcome prediction. And outcome prediction to begin with is pretty difficult for the reasons I think Sushi touched on a little bit.
Okay, thanks. Would anybody else like to address that?
I mean, I could venture a suggestion. I actually would take a step back, and I think the main challenge is a certain overpromising that's going around. By the way, anything I say, I am as guilty as anyone of. So this is not a criticism; it's a self criticism and realization. So there's a degree of over-optimism, I think, going on in all the literatures. The epidemiologic one, I think the computer science one, to the extent that I know it. And just this is a superficial example, but I think the over-optimism is built in the language. And so we already, I think, are talking about individual treatment effects, things that are mathematically unidentifiable, and so by definition inestimable. Okay, so if we go to our funders or the policymakers or the doctors and we tell, I'm going to give you individual treatment effects, I believe we are already committing to the impossible. And so, you know, I think once you take a step back, you realize that the problem is really about finding groups of people that somehow are different in their response to treatment from other groups of people. And then I think at the time we realized this, and again, I'm guilty of overpromising myself, in grants in particular, okay? I think that we take a second wrong turn, which is we experience a certain narrowing of thought and an overemphasis on toy problems. And so, again, you know, from things I've done, some of us will focus on predefined subgroups, and then we will claim victory because we have the efficient estimator in that problem. And we'll ignore all sorts of difficulties. I think Suchi mentioned missing data. We'll ignore the fact that the outcomes are censored. We'll ignore all sorts of complications and then claim a quick victory on the toy problem. Another way the narrowing of thought manifests is we will keep doing what we are each comfortable doing. And so some of us are comfortable doing big regression models, parametric ones, and I bet some of you are really confident doing crazy neural network models. And there is very little tying the approach taken to the actual definition of the problem. And so I think if I had to identify the overarching difficulty is the difficulty of talking to each other and maybe conceding that some things, I for one will immediately concede that the computer scientists are better at predicting stuff than I will ever be. And so how can we combine that skill with maybe the skill of another group in problem formulation and so on. So that's what I think is the hardest problem.
I think, thanks. I think one of the big issues I see is lack of an evaluation framework and the seriousness with which we evaluate. So I think both are important, not just a framework, but the seriousness and the commitment. So when I say lack of an evaluation framework, obviously in prediction, there's an easy way to evaluate as soon as you're trying to do something that is causal in nature. You have to be more creative about how you're going to evaluate. So there's one aspect of that, but the seriousness part I think is even more important. And the reason that what I feel we see a lot of is this desire to take a problem, come up with some formulation, use some flimsy metric for evaluating and declare victory. Then I think what we really need, what I found in practice is that in order to, if there's a real problem where you're very serious about using the estimated effect to do anything important, like you want to actually drive behavior change, you want to drive the uptake of that idea in order to change the way we practice, which would be pretty amazing. I think the gap between what we currently publish and declare victory on versus what it would take to actually get uptake is extraordinarily wide. And I think some of it has to do with, as you start evaluating it, you start learning about confounders you hadn't imagined, you start learning about factors you should be including that you're not thinking about. And also like some of the challenges I mentioned, like around data sparsity and the fact that we do discretization, et cetera, or the granularity at which we're modeling the problem itself, are things that I feel we discovered as we tried to take a solution set and close that gap and make it actually implemented in practice, where physicians would actually sit down and evaluate, and discovered that these gaps are very real. So anyway, those are what I see as real roadblocks. And I think in the scientific literature, I don't feel there's real appreciation for trying to close that gap. I think our current metrics in scientific publication is very much around what's a more fancy model you can add to the title of the paper. And ideally, it's like highly uncreative A cross B type work, adding a creative model to something that already exists and declaring that victory. And those papers get a lot of citations. And the papers that truly try to like push the boundary in terms of identifying where these gaps are in order to get uptake, I think those are much harder to write, much harder to produce, but I think much more important for the field to get credibility in the long run.
Great. I think that will lead me to a follow-up question, which you're kind of touching on. Do you think that in the end it'll be better data or better methods or better, tracking of what's going on, like understanding of the actual data generating process, that will matter for making results that translate?
I mean, my very vigorous nod definitely means, I think the word data generating process is a novel idea to many modelers, which is just shocking to me. But I think we need to understand the data generating process really, really well in order to be able to be successful. And whatever we don't understand, we need to think hard about doing sensitivity analysis one way or another, so that we understand what we know and what we don't know.
Great. I'll take a response on this question from everybody else too.
I think all three are important. I'm getting back to your earlier question as well. I did find in my first few years trying to tackle these sets of problems that lack of realistic benchmarks was a big problem for me. I could easily create some synthetic settings to test on, but how do I know that those are interesting? I remember, Isa, a few years ago, you were telling me, I thought that you were building a big benchmark of causal inference problems where there were analogous randomized controlled trials that had been performed where one could at least look at average treatment effect and presumably some subgroup analyses as well. I think there is no single method that's going to be good for inferring heterogeneous treatment effects, but what we could try to do as a community is try to think through, well, for particular types of data, for particular types of settings, what are good methods, and then try to evaluate them, combined with the things that Rajesh and Suchi just mentioned in terms of building in very good processes for working with domain experts and thinking through that data generating process and making sure there aren't gaping holes in one's analysis.
So I agree. All three of the things mentioned are critical. That's the way to victory, I guess, in the end. Perhaps that can be part of developing better methods, but I think it's one of us, domain experts, people who like the causal stuff, people who like, I don't know the formal names for these things, people who like the machine prediction things. We bring something different to the table. And so just to tie in with David Kent's previous point about this being a harder problem, heterogeneity being a harder problem even in the absence of confounding. I personally have heard David, from henceforth David K. I've heard David K say, but we kind of have a good feel that higher order interaction terms in our regressions are closer to zero, and big interactions, think of these as product terms in regressions, are very unlikely. Very similar in spirit to this is the realization that the heterogeneity functional, so if you will, the conditional average treatment effect, basically the expectation of the difference of the potential outcomes, if you will, for two treatments given covariates, just to have an informal definition, that object is probably much better behaved than, for example, the individual potential outcome regressions. And so I believe I learned that insight from David K. And so it's now the task of the statistical people to say, well, okay, now since we can impose some constraints on this object, since we have background beliefs about it, what are the optimal estimation methods? And I think then, the machine learning community would be giving us optimal ways of estimating various pieces of that bigger piece of the puzzle. And so, and that's already, by the way, having narrowed our thinking to a pretty narrow definition of heterogeneity. Okay.
We'll take a question from the audience that released this, which is like, what are the differences across medical conditions? Is there a place that one should start, like a particular subdomain?
So that sounds like maybe a question I should try to address. And, I'm not sure I have a great answer for it. I know that I've worked a lot in the cardiovascular space because that's where the data is. And that's the field I understand the best. And the databases tend to be very large. But more than, for example, what clinical area might benefit from this type of approach where you're looking at for heterogeneity of treatment effect, I think you have to look really at kind of the shape of the decision, and you want to make sure that it's going to be a worthwhile problem to approach. Not that it's oncology or cardiology or neurology, but you want to find a treatment that you're fairly confident works to begin with. You don't want to try to do exploratory heterogeneity of treatment effect analyses on things that might have a zero average effect, because you might well find something and it's probably not very credible. You probably want to look at treatments that have some cost or burden to them, because if a treatment is completely innocuous and inexpensive, then defaulting to the average treatment effect is going to be, a good approach. If it works, give it to everybody. So, so there should be some treatment-related harm or some cost or burden associated with the treatment. Ideally, the treatment-related harm and the cost or burden should be in some fine balance with the benefit. Because if the benefit is much, much greater than the cost of the harm, your model is never going to be good enough to select out the patients who really don't benefit. So we spend a lot of time thinking about kind of those things, whether the decision threshold and the average effect are in some close proximity to one another that makes it a worthwhile problem to go after. And then it has to obviously have, you know, public health relevance.
A quick follow-up to that, is there a difference across types of health systems or in like a community hospital versus a large metropolitan hospital?
I don't follow the line of questioning. Was the first question really around where do we think there are going to be the biggest immediate wins in studying heterogeneous treatment effects?
I think it's also like if someone is trying to get started, what area should they focus on? Not because of just the wins, but because of like ease of entry to the problem, well-defined questions, there are many other considerations there.
Thanks.
I think if you're looking to just get started and just get your feet wet and tackle the problem, I would say looking at problems in the, I mean, I don't think these are necessarily the most clinically impactful per se, but looking at problems in the ICU setting is a fine starting point. And, you know, with PhysioNet and Mimic, there's a large enough data set that's available. And I think there are plenty of really interesting and difficult methodological problems there that if you were just looking for both a large data set, a clean setup, and the ability to create enough of a sandbox to be able to ask questions, I think that's a very good starting point. And from the point of view of high impact clinical questions, I would even say there are some, I mean, end of life, there's so much heterogeneity in response in so, you know, across medications. So I would say maybe even clinically, there are some exciting, there are exciting opportunities there.
I'll give it a different, I'll add on to that, which is that I think in cancer, it's an interesting area to start to get into for these questions. Of course, there's a huge amount of existing work on precision medicine in cancer, which is all about identifying heterogeneous treatment effects. And so lots of literature to build upon and to ask about how one could build learning and inference algorithms that could attempt to, first of all, reproduce what we know is true, but secondly, try to go beyond it. And also excitingly, lots of open data sets that one can use, ranging from more sort of biological assays like the LINKS data set from the Broad Institute, which looks at the response of a number of different tissues to various drugs, including cancer drugs, to patient registry data. So I'll just put that out there as another one.
Great, thanks.
Just one point. I think it helps to think in two directions. And so there is the aspect of how important is the problem, which I think both Davids spoke to. I would ask people to work on cardiovascular disease and cancer because my family history suggests this is what will get me. So I have a stake. But another aspect is how hard is the problem. And you know, over there, everyone will have to decide whether they want to just get their feet wet or whether they want to solve a very important problem that will usually be hard. David's and Suzy mentioned many of the things that make problems harder, and data is one of them. I'd like to add that you need to have a sense of what your model is going to be used for. And so a very big challenge, I think, is when your model is going to be used in a population that is potentially different from the one you have access to massive data sets from. And that's for the obvious reason. The covariate distribution is different. I think you ML guys call this covariate shift. I'm trying to learn the lingo. But there's also a very clinical aspect to this, which is if you go to a new population, the distribution of treatments is going to be very different. And cancer, I think, is a minefield for that because there is a lot of variation, and the treatments tend to be very different and very dependent on tumor characteristics like mutations and other genetic, genomic, omic stuff. And so, once you think about targeting a new place, the causal considerations that Suzy has been emphasizing become critical, because basically you can not learn about your model, and you can definitely not evaluating without considering how the covariate distribution changes, how the post-treatment covariate distribution changes, and how the treatment varies over time. And so these, I think, are very exciting and perhaps the most clinically relevant kind of areas, but are probably also the hardest.
Great. So three of you said that like understanding the data generating process is important for making progress. Right now we have an ongoing pandemic for COVID-19. There are a lot of things that we don't understand. Lots of people are trying various stuff. There's shifting populations. Like what role, if any, do you think like heterogeneous treatment effect estimation has for the current pandemic?
I'd love to take that one first. So we've been neck deep in COVID data for the last like 3-4 months. So I think the answer is we don't really know. I think there's so little that is really known about COVID from the point of view of, there are so many open questions, like first question, even something as simple as, providers are contending with, is COVID just like acute respiratory failure, or is it a little bit different? And if it's different, is it that it's overlapping or totally different? And what part of it is new and why is it new? And then second is they're trying all sorts of treatments. And since March, there's been a very aggressive iteration on the treatment protocols that different systems have been offering, starting from, in the beginning, the belief was people were very worried about using machines like CPAP, BiPAP, because they were aerosol generating. So the thought was, let's aggressively ventilate patients. And then at some point, like even if they need a little bit more than 6 liters of oxygen, let's put them on a ventilator after that. And then at some point that changed, and there's increasingly anxiety in the community that aggressive intubation is leading to mortality. And even our understanding of what and is proning effective or not. So I think probably the answer is, for some people, proning is effective in general. Maybe for some people, intubation is worse than others. But who's responsive to what is something people don't really understand. And I think a perfect place where more careful use of this kind of data and estimating treatment effects and methods for estimating treatment effects and heterogeneous treatment effects could be very useful and productive. And then there's another question, which is the degree to which it can be estimated, given the data is so wildly messy, because people are changing things all over the place. And I think there, I feel like the answer is less of whatever I've estimated is 100% correct, and more a version of the degree to which I can trust it. And I think that has to, you have to iterate on that. Like, so you have to ask questions in the beginning that are a little bit coarse, like, can I rule out things? Can I rule in things? And then maybe once you can do rule out, rule in, maybe you can refine and do a more careful prospective data study that allows you to drive the policy that's, to influence the policy that's driving the data itself, collection itself, in order to come up with more precise estimates.
I think there's a nice analogy actually between the cancer story we were just talking about in COVID, and that treatment is changing so rapidly across time. So there's a very interesting methodological question. If you're using methods that are based on comparators, the comparators are potentially changing across time, and how do you sort of bring together the observational data from such different distributions in some sense together? You see that? I think we'll see that in both cases.
What about at the level of actually getting the infection itself? Like some people get sick, some people don't. Do you think like methods like that we're talking about would have a role?
With the right data.
Yeah, I think part of the challenge in COVID is that we don't even know if we're measuring all the things that are necessary to differentiate between the different outcomes people are experiencing, right? Including different treatment effects. And I think we're rapidly discovering, like the examples with the autopsies that showed clotting as one example, which then shed light on new mechanisms, is very exciting. One thing that's really cool about, I mean, there's nothing cool about COVID, first of all, so that's not really the right way to say it. But as a data scientist, I think it's very historic to be part of a time, obviously beyond it being a pandemic, but also how you're literally in the midst of something where you barely understand it. You're collecting all this data, and it's not just a nice to have, but a critical need that we need to use this data to rapidly understand the disease so we can really inform treatment strategies. And I think that's just unprecedented. And the speed with which we can do this is very critical. And obviously the clarity with which we can do it is also important, because if you come up with the wrong conclusions, as we saw in some cases, people are very willing right now to listen. So you can scale up wrong ideas, and that hurts everybody too.
David, you mentioned, David Santa, you mentioned something which is like with the right data. And like somehow like one would believe you, or believe anybody in general that said that if we had like magically the right data that was very good, then things become a lot easier. Suchi, you also mentioned that there's this like trade-off, like a willingness to like accept an answer that is not good and try to iterate on that. How do these things play together when we don't know what the right data is? Like that's part of the question.
Where I've been moving some of my lab's research has been toward data types where you have so much more granularity that there's a good chance that there'll be some proxy for the data you might want to have within it. And for that to be the case, you are almost certainly going to have to move beyond the type of clinical data that at least my lab has been traditionally working with. And so we've been moving much more to untargeted proteomics, metabolomics, and things of that sort.
I think right now, one way we do research is we take a data set, we have a fixed problem, and then we produce a result. But to me, it increasingly feels like you have to understand enough about the problem you're solving, or be partnering with people who understand a lot about the problem to be solving, and then having more of an inquisitive, interactive process where you may come up with a first cut of something and you have to really probe and query the data and do tests to ask these questions, like hypothesis, like, is the issue that we're not measuring the right factors? Is the issue that whatever we've estimated is incorrect? Is the issue that our methods are just bad because, there are types of issues like biases or confounding or like issues in the data that are hurting the quality of the estimates we're generating? And I think these are all like, like a little bit of an investigative process in trying to get to a solution set in the long run. And in a way, this links to experimental design, right? Because as we start taking this kind of a mindset and we partner with our data generators and our, like in this case, clinicians, then it's possible for us to also inform practice, but also inform like what they record or what data is getting collected, which is a little bit more, I think, end-to-end thinking than typically in data science, where we sort of assume a data set's given to us and a question's given to us, and our job is to just sort of run the crank.
Suchi, I really like that framing. And I think it connects also to what I was just saying a second ago. It's that you have to go beyond just, of course, the untargeted metabolomics and proteomics to think about other factors. And so I think in order to enable that vision that you nicely outlined, we do need to have proxies of the things that we want to try to elicit available in some data, so that we can design algorithms to, in some sense, suggest the questions that we can take with us as part of this experimental design process. And then get better data based on the results.
A question from the audience relates to this is like, how does this fit into like the academic pipeline? You know, if you like have like small bits of living work that you're iterating on, how does it fit in compared to the typical, like you have a data set, you produce a result, it's better than other results?
The question is, do you want to do science or do you want to publish papers?
I think that this is a question from somebody who's a student potentially, and you know, papers are part of their job, right? And you know, I think
Actually,
that if you,
I think maybe the way to answer that is there are methods papers and there are discovery papers. And the challenge is if you're only looking at your peers who only publish methods papers, then you'll be forever on the hunt. So I think in order to do this successfully, you need both discovery papers and methods papers, where discovery papers are eventually about the big scientific discovery that was made by leveraging one or more cycles of this process. And then methods papers are along the way gaps that you found that you then were able to close. But to me, meaningfully, this approach leads to both more meaningful science papers and more meaningful methods papers than the reverse, where you don't have a problem in mind and you're in the hunt for a paper, which is historically what I felt is most commonly the approach people pursue in a community. And I don't feel like those papers, I mean, maybe they get a lot of citations because you have the right words in the title, but I don't think they like stand the test of time as much.
It does seem that
Was that a more palatable answer?
yeah, probably.
But Issa, what were you gonna say?
Time.
I think Issa was gonna say something.
I was going to say, yeah, I mean, that's a nice framing. I think COVID, you would be jumping in the middle of a hot scientific mess. I personally have been reluctant, or at least more circumspect in branching out, because infectious disease is not within my circle of competence. And so you'd be jumping into a scientific problem. It's definitely a very different environment where, you know, basically you have to define the problem for yourself, and you have to convince domain experts that that's the correct formulation, the formulation that maybe formalizes their thinking, and then you would have all the data challenges. I think there is a lot of data and it's being shared widely, which is an improvement from other domains. But I think the data law, I think what Shuchi calls the data generating mechanism, I think even the observed data law is very adversarial for COVID. And so it's not a area rife with toy examples, it's serious science.
Changing gears a little bit, what about evaluation? We're talking about like building data sets where we have like standard benchmarks. Like, what are the right metrics we should be looking at, specifically both in terms of like the quality of the data to know if we're doing a good job, and in terms of the actual effect estimates?
Yeah, I think the field is just developing these frameworks now. I don't know, I'd be interested in what other people think on this, but, you know, generally, you know, you can't measure the accuracy of prediction in individuals. You have to measure them in groups, and, you know, there, I guess, some new metrics that have been proposed for how to convert conventional metrics that we use for just outcome prediction in terms of discrimination for benefit, benefit concentration, I think is based on Lorenz type curves that some folks in Vancouver have toyed with. So there's discrimination, there's calibration in terms of statistical accuracy and what's that decomposed to. And then there's net benefit. And those are kind of the three areas that we look at with outcome prediction, at least in my field. And I think of all those, net benefit is perhaps the most important and the most neglected. What you really want to know is your prediction good enough to improve decision making? And we've found, and when we start looking at this and how the models transport, is that more often than not, you will often disimprove prediction by using a model in a new system than you will by using just the best average effect. We're still thinking about the best ways to evaluate models. That's one of the areas we're quite interested in.
For my question about the data, sometimes the knowledge that's needed to know if there's something weird in your data or not is not necessarily available to a computer. Meaning you're like the electronic health record didn't process this variable for this subset of patients. It's not necessarily recorded, but it would be useful for methods to do that. Do you think that this is like a thing that there's willingness to do? Of course, it's possible to do, like in your experience. Like really tracking data provenance. I want to be more precise with that.
Right. What is the incentive mechanism? Right. That's what I wonder about.
You can think about it as it's like a promise. Like you give me the data provenance, I give you better answers. Well, you haven't given me the better answers. Well, why should I spend the money to figure out how to track the provenance, which is a nuisance?
What do you mean by data provenance, Rajesh? Can you clarify?
I really mean like sometimes like you can talk to a bunch of people and figure out, for example, like why like BMI is missing. And like if you talk to enough people, you can get the answer to that. But like there's no way that's recorded in like a digital place or something that's easily accessible. It slows down our work. It can lead to errors if we don't know these things. And you mentioned it yourself, like, as you go and talk, you uncover things that you talk. But sometimes they're just like known things. There's somebody who like built the data processing pipeline. Who knows it? And it's something I've experienced in my own work too, with BMI. So I'm picking on it.
When the Precision Medicine Initiative, now called All of Us, was started, there were a number of workshops with tens to hundreds of people at them, where there was widespread discussion about, okay, these are the big picture problems we want to tackle. What type of data do we need to gather in order to be able to answer these clinical and operational questions? And so the data was at the, that very much drove, as far as, I was an outsider watching in, that seemed to drive very much the data collection setup of the process. And I imagine the same will happen in many other areas.
But in that, do you know if they track down to like smaller issues that are like particular to each individual health record, or is it like surfacing health record data?
I think with existing health records, there's only so much you can do. I believe it was more about the auxiliary data they were collecting after, in addition to it.
I think it's a really good question. I feel like right now, there isn't really, physicians are so busy. So from their standpoint, asking to do more light data collection on rationale for why something is getting collected or not collected seems pretty hard to imagine. So the question is, But if we had that, it would make the data so much more useful or so much more reliable. So the question is there a way to collect that non-invasively? And if we can't collect, I feel like that would be worth thinking about. Is there a way to collect that non-invasively or collect it in a much less expensive way, which is maybe it doesn't try to ask that question every time. It only asks that question when it's most useful to ask, which is 95% of the time we know what they're doing. And the 5% of the time when they're behaving in a way that is unexpected, there's an opportunity to ask the question like, are you not ordering this because, or are you ordering this because? And that might be a way to improve data provenance dramatically. The other thing is, on the flip side, us becoming more aware and intelligent about it, this is going to sound really silly, but one of the things I've discovered doing research in this field is you have to be, this is going to sound super silly, but a hustler. Like, you've got to be willing to go learn about what it is you need to learn from the people around you. And there are some easy ways to learn it, but it certainly isn't simple or clean. And so, you know, part of that is just having a couple of collaborators. Like, you can get 95% of the way very quickly if you have the right collaborators, you have the right communication framework, and it feels very daunting in year one. But I actually think it's not that hard over time. It becomes much more, I think it's much harder when you're starting. It feels more overwhelming, but my sense is after you've done one or two projects, it becomes very natural and easy.
Yeah, we're just about out of time. I'd like to have everybody give a few closing remarks on like what you think about, you know, the direction that we as a community should be pursuing here. We can go in any order who wants to go first.
Maybe we can go in the order of introductions, just to make it simple.
Sounds good. David Sonde, take it away.
I've been super happy about the machine learning community's growing interest in causal inference. And we see this in a growing number of papers at NeurIPS, ICML, Machine Learning for Healthcare conference, and so on. So I think that's really promising. It speaks well for our field. I think we need to, you know, one of the trends I see happening is that folks are starting to move away from the basic questions towards more complex questions of relaxing assumptions and so on. And that's really good. I myself am starting to move now toward even the questions that come after those, which are to draw on Madeline's talk from earlier today about integration. So how does one integrate inferences about heterogeneous treatment effects into clinical practice? And that's where I see some of what will be some of the, I think, the most interesting questions in the next few years.
So I think I'm next. So I guess I started off really pessimistic, and so maybe I'll end a little bit more optimistic. I think that you know, I think if you look at the progress we've made over the last couple of decades, I think we've made just so much progress in terms of how we think about evidence and how we think about heterogeneous treatment effects, and just the fact that the whole machine learning community is interested in it. When people said heterogeneous treatment effects, even just a decade ago, people would really think of what we called one variable at a time subgroup analysis, males versus females, old versus young, with diabetes versus without diabetes, and so forth. And now people are really thinking of prediction of treatment effects given all a person's relevant covariates, which is, you know, progress. I think tremendous progress. And the other place I see a lot of progress is in, and the place I think we need more progress is really in the data infrastructure. You know, these things just didn't exist. I mean, when I, you know, was practicing even a decade or two ago, I would spend half my day just looking, you know, for the chart, the loose leaf binder with the chart in it. because the patient was in radiology or something like that. Now I can do my notes from anywhere, and everything's automatically captured. And now we have these databases that have 100 million covered lives. And ultimately, approaching heterogeneous treatment effect is going to require these very, very large databases. And we're already learning about how to, compile these databases. I was going to say one more thing, but maybe, thankfully, I can't remember what that is, so I'll just pass the mic.
Yes, I believe next.
I think the future is going to be great. I'm not too worried about the data, even in America, with crazy fragmentation of the healthcare system. In the end, data will be available from millions. I think the next step would be to work with the domain experts, define the problem carefully. By defining the problem, I mean be able to tell yourself why you need to estimate heterogeneous treatment effects. Is it to personalize decisions? Is it to transport average effects to a new population? Is it to learn about the variables that drive the heterogeneity? Is it for some sort of counterfactual prediction and so on? We could go on with a list. Once you do that, I think there's going to be fantastic opportunities for future research on the optimal methods for each of these different sub-problems within the big heterogeneity umbrella. That's it.
I want to just second what Isa just said. I think in one of the things that's super exciting, or what I find different about this domain, is there are both problems everywhere, and any single problem is very complex. So rather than, I think historically in machine learning, there's been sort of desire to, here's a new idea, here's a new idea, here's a new idea, here's a new idea, and flag plant and write lots of papers on lots of different things. And I personally think one of the super exciting things about this area is that any one problem, you can go deep. Like you can keep iterating until you go deep. And so I personally agree with what Issa said. Find other people who can be good collaborators, find other labs at this intersection, work with them. Even if you start with one problem, there's just so much open greenfield, like there's so much room for work that you just pick one and then dive deep and just iterate from there. Like literally any of the topics mentioned today, I mean, they're all open areas.
Great. I want to thank everyone for coming, and I want to thank the panelists for a wonderful panel. After this, we'll have a wrap-up session. There's a Zoom link in the chat. You can follow it there, or also follow it on our website in the agenda. Thanks.
Thank you, Rajesh.
Thank you.
Thank you.
Bye, everyone.
Bye.